Hello, my name is Jennifer Alonso. I am a student in the microbiology lab course this semester, and my instructor is Lisa Friend. This video is going to explain the purpose and procedure of pure cultures. So firstly, a pure culture is a type of culture that is going to show only growth of one particular organism or bacteria. A pure culture is derived from a mixed culture, which all cultures are at first, because there are several different types of bacteria in them. So in order to get a pure culture, you must go through the pure culturing procedure of a bacterial specimen. In order to do that, you need a few different items. You need 2 streak plates, which have different types of agar. There's a TSA agar, which is supportive. It's a supportive agar, which means that it is a good environment to grow all different types of bacteria. It's pretty widely used for that reason. And then there is EMB agar, which is selective and differential. It is selective for E. coli bacteria and differentiates for lactose fermentation. This agar is used for E. coli bacteria, which is a common bacteria to find in a urine specimen due to urinary tract infections. Something that makes the E. coli bacteria stick out on the streak plate of EMB agar is that it will turn a green metallic color. So you have your 2 streak plates with the 2 different types of agar. You have a Bunsen burner used for sterilization of the inoculating loop, and to keep your procedure aseptic and without any introduction of different bacterias during the procedure. So firstly, what you will do is you will take your inoculating loop and sterilize it with the Bunsen burner. You will, at that point, dip it into the bacteria culture, and you will spread it on the first plate. You will spread it in a back and forth motion for the first 1/3 of the plate. After that, you will sterilize the loop. You will rotate the plate 90 degrees, and you will drag the inoculating loop that has been sterilized through one part of the initial streaking line. And then you will continue on to do the back and forth movement for the next 1/3 of the plate. After that, you will sterilize the inoculating loop again, rotate the plate 90 degrees, and you will drag the sterilized loop through one part of the second streaking line and complete the streaking for the final 1/3 of the plate. This is going to dilute your bacteria sample with each streaking, and that's what is going to give you your isolated colony in order for identification. The desired outcome of the streak plate is minimizing the bacteria colonies so that it can be isolated and identified for treatment. It's important to use a clinical specimen in the pure culture procedure in order to do just that, to isolate the organism that is predominant in the specimen so that adequate treatment can be prescribed to the patient based on the antibiotic sensitivity of that bacteria that has been isolated and identified. After the streaking, I do want to mention that it goes into an incubator in the lab, and the results are analyzed the next day. And at that point, after analyzation takes place, the patient is able to be prescribed a medication that will be able to kill the bacteria. Thank you. Bye.